Role of insulin in carbohydrate metabolism. Now, insulin is a polypeptide hormone. Okay, it is secreted by beta cells of islets of Langerhans. Now, insulin favors the synthesis of glycogen, triacylglycerols, and proteins. So, glycogen, triacylglycerols, and proteins are being synthesized in the presence of insulin. Now, this insulin is composed of 51 amino acids, and this 51 amino acids of the insulin are arranged in two polypeptide chains, and these two polypeptide chains of insulin are referred to as A chain and B chain, and these two polypeptide chains of insulin are linked by two disulfide bridges. So there are two disulfide bridges between this A chain and B chain of the insulin, which is composed of 51 amino acids. Now, insulin reduces the glucose concentration in the blood by stimulating the conversion of glucose into fats or proteins or ribulose 5-phosphate or glycogen. So the conversion of glucose into this thing is stimulated by the presence of insulin, and that is how insulin is playing a role in reducing the glucose concentration into the blood. Now, in liver, insulin inhibits these two processes, that is gluconeogenesis and glycogenolysis. Okay, gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors. So the synthesis of glucose is inhibited here, and in glycogenolysis, glycogen is broken down and glucose is produced. So both the processes are producing glucose, and this is inhibited by insulin, and that is how insulin is reducing the glucose concentration in liver by inhibiting these two processes. Now, in muscle cells and liver cells, insulin increases glycogen synthesis. Okay, insulin stimulates the conversion of the glucose into the glycogen. Now, in muscle cells, insulin increases the glucose uptake. If there is more glucose uptake, more glucose is available for glycogen synthesis. So insulin increases glucose uptake. Now, how does it do that? Okay, it increases the number of glucose transporter in the cell membrane. So in cell membrane, you have more glucose transporter, and that is the reason why the muscle cell is able to uptake glucose more. Okay, there is increase in glucose uptake because there is increase in the number of glucose transporter in the cell membrane, and this thing is happening because of the presence of insulin in the muscle cell. Now, this epinephrine and glucagon, these two hormones have opposite effect on the insulin. Now, both of them act to increase the glucose concentration in the blood. Okay, insulin reduces the glucose concentration, whereas both of this increases the glucose concentration. Okay, and that is the reason why these two have opposite effect to the insulin, that is opposite effect with respect to insulin. Insulin just reduces the glucose concentration, whereas epinephrine and glucagon increases the glucose concentration. Now, this epinephrine and glucagon stimulate the conversion of fat, glycogen, and pyruvate into the glucose. Okay, this is stimulated by epinephrine and glucagon. So insulin converts glucose into these things, and epinephrine and glucagon converts these things into glucose. Okay, so they have opposite effect with respect to each other. So insulin stimulate glycogenesis and glycolysis. Okay, glycogenesis means synthesis of glycogen, that is the use of glucose, and glycolysis means the breakdown of glucose. So in both the processes, the concentration of glucose is reducing, and insulin stimulate that, the reduction of concentration of glucose, whereas glucagon or epinephrine stimulate glycogenolysis or gluconeogenesis, that is breakdown of glycogen into glucose and synthesis of glucose from the non-carbohydrate precursors. So in both of the case, glucose is forming, and that is the reason why these two are increasing the glucose level, the glucose concentration, and insulin is reducing the glucose concentration. Now this insulin also inhibits the glycogenolysis or gluconeogenesis. Okay, that is the formation of glucose is inhibited, and that is how there will be reduction in the glucose concentration. And glucagon or epinephrine inhibits the glycolysis or glycogenesis, that is the utilization of glucose. Okay, that is inhibited by these two things, and that is the reason why there is no reduction in the glucose concentration, because of the fact that this process will not take place. And in this case there will be no increase in the concentration of glucose because these two steps are not taking place. Now let's read about glycogen storage disease. Now this glycogen storage disease occurs because of the genetic deficiency of the enzymes that are involved in the glycogen metabolism. Okay, there is genetic deficiency with respect to these enzymes, and that causes glycogen storage disease. Now there are many diseases which result due to inherited deficiency of these enzymes involved in glycogen metabolism. For example, there is this disease, von Gieker's disease, which is the glycogen storage disease, and that is caused by the deficiency of liver glucose 6-phosphatase. Okay, so there is deficiency of glucose 6-phosphatase in the liver, and that causes this von Gieker's disease. Now this Pompe's disease, this is another glycogen storage disease which is caused by the deficiency of lysosomal alpha-1,4 and alpha-1,6 glucosidase enzyme, that is acid maltase. Okay, so there is deficiency of this enzyme which results into this disease. Now this Hirsch's disease occur when there is deficiency of liver phosphorylase. Now this Taury's disease occurs when there is deficiency of muscle or erythrocyte phosphofructokinase 1. Okay, if in your muscle cells or in your erythrocytes, if there is deficiency of phosphofructokinase 1, then this Taury's disease will occur, which is another glycogen storage disease. Now this Mac-Urdley's disease is another glycogen storage disease which is caused by the deficiency of muscle glycogen phosphorylase. So in your muscle cells, if you have deficiency of glycogen phosphorylase, then Mac-Urdley's disease will occur. And at last you have Anderson disease, which is caused by the deficiency of branching enzyme. Okay, so this is the list of enzyme. Because of the deficiency of these enzymes, these diseases are caused.